professor in Department of Metallurgical and Materials Engineering, IIT Roorkee. She had played an important role in the creation and growth of the Center of Nanotechnology. She was an active researcher in diverse areas of material science and technology. Her major research interests were development of various novel composites and nanomaterials for radar and microwave observer application, e-waste management, and nanotechnology for water purification. Now, let me give a brief introduction about today's speaker, Professor Ali Kadam Hassani. He received his PhD in bioengineering from MIT 2005 and Master of Science and Bachelor of Science from University of Toronto, both in chemical engineering. Currently, he is the CEO and founding director at the Terasaki Institute for Biomedical Innovation. Previously, he was a professor of bioengineering, chemical engineering, and radiology at the University of California, Los Angeles. He joined UCLA as the Levy Knight Chair in November 2017 from Harvard University, where he was a professor at Harvard Medical School and faculty at the Harvard MIT Division of Health Science and Technology, and as well as associate faculty at the Weiss Institute for Biologically Inspired Engineering. At Harvard University, he directed the Biomaterials Innovation Research Center, a leading initiative in making engineered biomaterials. He is an associate editor for ACS Nano. He served as the research highlight editor for the Lab on a Chip. He is a fellow of various prestigious academic institutes, including American Institute of Medical and Biological Engineering, Biomedical Engineering Society, Royal Society of Chemistry, Biomaterials Science and Engineering, Materials Research Society, Nanosmart Society, and American Association for the Advancement of Science. He is an author of more than 650 research articles, editorial and review papers, and more than 70 book chapters or edited books. And he has more than 50 patents and patent applications. And he has been cited more than 82,000 times. And he has an H-index of 146. And he has also founded 2 companies. Okay. With this brief introduction, now I request Professor Ali Kadam Hassani to deliver his talk. Professor Ali, so now.
Thank you very much. Thank you. Appreciate. Thank you. You hear me okay? Awesome. I think sounds great. All right. So I really appreciate the invitation and love to tell you a little bit about what we're doing. It's around 6 a.m. here in Los Angeles. So obviously a little early. So if you don't mind, I'm not fully presentable, so I'll turn off my camera for the duration of the talk. But it's a great honor. I've actually never been to India. It's one of the things that I at some point hope to do, and hopefully when things settle down, I will have more freedom to visit. Today I'm going to tell you about our work, particularly in the area of applying engineering principles and various types of technologies, including nanotechnologies, to medical applications. These are my conflicts, as it was mentioned. And before I start, I wanted to kind of give you a little bit about the institute that we have here in Los Angeles. So the institute is named after Dr. Paul Terasaki, which is shown here. He was a academic entrepreneur. He developed a technology to be able to match donor and recipients based on basically cell surface markers such as human leukocyte antigens, which is a type of cell surface receptor. And this really enabled the widespread use of transplantation, which took this field to stages where it can be used in mainstream therapy. So he actually started a company from this initiative that that is the basis of the foundation and the endowment for the institute. So we're located here in Los Angeles, and this is one of our number 3rd building, which is a recent building. And our goal is to not only do and develop science, but also to foster the scientific discoveries that we make to practical solutions. And we are focused in this whole area of personalized medicine. The kind of work that we do involves a number of different things. And I think soon you'll see the diversity of the types of research that we do. But one of the things that we do is we make a lot of biomaterials, including a range of different nanoscale materials. For example, being able to make surgical materials. These would be materials that can be used in the body, for example, to seal wounds, or be able to be delivered through blood vessels to fill aneurysms or block internal bleeding. So these are kinds of things that we've done quite a bit on, and some of these are now actually in people as well. The other thing that we do a lot in the lab is to actually develop devices. These are devices that are integrated with electronics and other sorts of sensors. And the whole idea is that these devices will not be passive participants, but also actively participate in the healing or the therapeutic application. And as you can see, these devices often have um, lots of things like electronics and sensing components embedded inside them. We also work a lot on cells of different sorts. These are both stem cells and immune cells that can be tailored to an individual person's need. And what we can do with these is that we can use them for a range of different applications. So for today, I'm going to not talk about the top things on top, but focus especially on the two of the things at the bottom, which is making implants and devices that could be useful in different types of applications, particularly making tissues using technologies like 3D printing, and alternatively, to be able to also use those tissues in making what we call organs on a chip or tissues on a chip, the so-called personalized microphysiological models. These would be models that can be used to basically mimic human physiology outside of the person. So I think these are the kind of things that we do, which I think has a lot of us excited about the opportunity here. And as you see here, this can only be done by using a lot of different expertise from many different types of disciplines. And finally, we work also on personalized nutrition, which is an area where we try to develop nutritional solutions that can actually enhance the health of people as well. So let's start off with this whole area of personalized microphysiological models. And if there's time at the end, we'll talk about some of the things about our bioprinting advances. So as many of you guys know, and particularly during the pandemic, drug development and vaccine development is a very important process. And typically it takes a lot of money and many different, a long time. And it's in many ways, it's like a funnel. You start off with lots of different compounds and you go through different stages of selection to identify the one that's going to be your approved drug. And as you go through this process, the costs continue to expand significantly. And finally, if you're going through the years, that you see that these processes could take many, many different years. So the process involves basically first going through lots of tissue culture dishes, then animal models, then different stages of human clinical trials. And if you're failing at a stage like this clinical trials in this second or third levels of clinical trials, you've already spent literally hundreds of millions of dollars in the process to develop these compounds. So it's really important that we try to identify which drugs are going to be safe and efficacious for people right from the beginning. So and this is the reason why when you're looking at pharma companies, the R&D budget of these companies is really astronomical. So why do drugs fail? Well, when you look at the reasons why drugs fail, you see two major reasons. One is our inability to predict their efficacy, and then the other one is our inability to predict their safety. And really that is the major issue, and this is why drug development is so expensive. And then the other aspect is really what we call the aspects relative to personalized medicine. even if you get a drug approved, every single person behaves somewhat differently. And that's why it's important to kind of understand this different behaviors of different people. And these are the reasons why you have drug recalls, like Vioxx, which was a drug that actually caused a number of different issues and had to be recalled with a lot of concerns. So the goal really is to eliminate the use of tissue culture dishes and animal models because they don't really mimic the human physiology. So that's kind of where things stand. And when you think about how to do this, there's been a concept that has emerged over the past few years that's basically relies on making tissue engineering principles to develop different types of tissues for actually making organs and tissues for transplantation. So these are some examples of this. You can see that basically, if you can make tissues from human cells, you can potentially even interconnect these tissues to get more of a systems behavior, which is representative of how a human body performs. And you can have these in various sorts of things. You can have barrier tissues like lung, gut, and skin. You can have metabolic tissues like liver and other types of structural or cardiovascular tissues like bone and heart, etc. So this is the idea. And the idea is that one day we'll be able to have person-specific cells and then be able to identify how that individual person will respond to a particular drug or compound to minimize issues with drug toxicity, as well as being able to work with other things like being able to address issues such as um, how individuals respond. So there's been a lot of work in this field over the past few years. These are some examples of them where the idea has been to take standard technology, which typically relies on putting cells in a 2D monolayer and often uses animal cells and it's restricted to individual cell and tissue types, to models that basically are using three-dimensional tissue culture, uses primary human cells or induced pluripotent stem cells, and uses fluidic systems to be able to control how we move liquids around between these different tissues. And virtually every tissue has now been modeled using these types of technologies. And when you look at these, you start seeing some general trends. For example, you see that basically there is an appreciation of not only using the human cells, but also the different cell types that makes the tissue. There's appreciation of the microarchitecture of the tissue, for example, the villi in the intestine or the vascular structures. And there's appreciation of the forces and stimulations that are related to that tissue. For example, using the shear forces of blood vessels or the deformation, the elastic aspects of the lung and its mechanical deformation. Of course, lots of other biochemical signals. So we've been working in this area for a few years, and these are some of our early prototypes. This is an example of a system that is used to make what we call a tissue on a chip. What's inside this box is actually where the cells and tissues are housed. And in the surrounding is basically the types of systems you need to monitor what is happening inside these systems. These include things like different types of sensors and displays to be able to visualize things, as well as different types of valving and pumping, like these pneumatic systems, to be able to control movement of the fluids and close and open valves as needed. So if you zoom into this box, what you see are basically modular platforms. Here you can see that these modules, we have a number of different modules based on the kind of things that we want to do to these systems. So for example, we can have a modules that have the different tissue types inside them. These could be like almost like organs. We can have modules that control how the fluids are moving. So these could be pumps, reservoirs, or components that remove bubbles, like what you see in the top here in the video. Or you can have also sensing modules. These would be modules that can sense both biochemical aspects of what's happening, like, for example, concentrations of different molecules inside the solution, as well as being able to sense other things like other physical chemical properties. And when we do this, we start having systems that kind of look like this. And just to show you how we can have very fine control over these systems, here's an example of a video where you can see in real time these colors changing that shows basically how much control we have over our fluidics. And the way we get that control is through a central module here called the breadboard that basically is our controller. It allows us to open and close particular channels and be able to move and route liquids to different modules here. So this is how the system works. And basically what we can do now is to design the different modules. I'll show you this one of the modules here. This is like the actual bioreactor module. These microreactors are very small chambers that you can open and close and be able to deposit your cells and tissues inside them. What you can do is that you can make different types of tissue types. I'm going to show a couple examples, liver and heart tissue types, and you can use different technologies to put them inside these microreactors. You can use techniques like 3-dimensional bioprinting to do that, or you can use different types of patterning like light patterning or surface alignment to get the cells to kind of organize themselves in the way that it deems appropriate. And we've done a lot of work on this in different aspects. So one of the things that you want to do with these types of tissues is to show that the cells are alive and and also functional. And here's some examples. So one example here is to actually make liver tissue modules. Liver is a tissue that has a lot of regenerative capability in the body, but once you take it out of the body, these liver cells can't really survive for long. So to make them live longer, we mix the liver cells, which are these hepatocytes, with other support cells like stellate cells or endothelial cells or Kupffer cells. And this basically allows us to get the right kind of signals to these liver cells so that they can remain functional. And we make these little spheroids or microtissues that has these different cell types. And we can combine basically these cells, and we can grow these tissues that basically have a lot of the different enzymes and other types of characteristics of the liver. And they remain viable, and over long periods of time, they can maintain their viability. Now, when you make a liver tissue, it's important to see how functional it is. And one way is, of course, to look at the different markers or enzymes that the liver has. But the other way is to actually challenge the liver and see how it functions. So here's an example of adding a couple different compounds that are shown to be toxic in the liver, and they were originally approved as drugs and then subsequently had to be recalled due to liver toxicity. And we basically can mimic these behaviors at lower concentrations. We have less of these red cells, which are indicating dead or dying cells. And as we start adding our drugs, we can have more and more of these types of dead cells, showing that we have predictiveness. We can do the same thing with other tissues, and this is an example of the heart tissue, where you can see that with the heart tissue, we can't really take biopsies from people. So it's because heart doesn't regenerate as fast. And so what we have to do is we use stem cell technologies to be able to do that. So we can take skin cells or blood cells and be able to revert them back to a primitive stage and then differentiate them to heart cells. And by doing that, now we can actually have these micro tissues that mimic lots of aspects of heart tissue functionality. They live over long periods of time. They beat, as you can see here, they beat very basically in an organized way, in very regular intervals. And also they have the right kind of microarchitecture associated with heart cells. You can see the striation and patterns that are associated with heart cells. And when we challenge them with 3 different drugs that were originally approved and then subsequently recalled due to heart toxicity, we basically get the behaviors that we want. So as we add these drugs, we basically, you see more and more of these red cells, which indicates that the cells are sensing toxicity. And also when you look at beating rates, these spontaneously beat. And when you look at the beating rates, as you start increasing the concentrations of the drugs, the beating rates suddenly fall. So what I've shown you so far is basically making healthy tissues, but there's also models of making diseases. And I'm going to show you one example of a genetic disease that we've been able to model in our systems. So this is a disease that's called Progeria. It's basically early aging syndrome. So these kids that have this particular mutation wind up aging rapidly, and actually they die fairly quickly in their teens. And the reason for the disease is a mutation that causes basically an issue with the post-translational modification, that once the protein is formed, it can't get cleaved as it normally does in the mutated version. And this altered protein builds up in the cells and causes inflammation, which results in this rapid aging phenotype. So what we've done, one of the issues with all of this is that this rapid aging phenotype causes a lot of cardiovascular issues. So the blood vessels in these kids actually winds up stiffening, kind of like old age, and often the kids die because of this. So we wanted to model this, and for the blood vessels, the cells that matter a lot for this inflammation behavior is basically cells that are called smooth muscle cells. And we wanted to kind of model the disease by taking smooth muscle cells that are derived from healthy people, and also compare them to smooth muscle cells that are derived from progeria patients, and create a system in which you can actually develop these kinds of technologies. So we can develop microfluidic systems that actually do that. And to mimic the behavior of blood vessels, what we can do, normally in blood vessels, of course, every time the heart pumps, there's a mechanical dilation. So you have this natural cyclical movement and stretching of the blood vessels. And that's very important for what the smooth muscle cells see. So what we can do is that we can create these microfluidic systems that mimic that. So it's made from 2 different chambers. In the top chamber, you can have the cells and the medium. And in the bottom layer, you can have basically air to which you can apply vacuum. And as you apply the vacuum, then there's a stretching here that mimics what the cells see in their natural environment. And you can control all of this basically as you start increasing the pressure, the vacuum suction in the bottom channel, you can actually change the degree of deformation. And you can actually do this to different degrees. And here you can see some examples of how you can actually, the cells and their nucleus actually deforms when you're applying this mechanical deformation. So I want to show you how this actually affects the how the cells actually behave. And as I mentioned, when you have progeria cells, progeria smooth muscle cells, they have a inflamed behavior. They basically release molecules that are associated with inflamed tissue. So the first thing we wanted to see is how the cells in these biomechanically active systems behave compared to a standard static Petri dish. And one of the things that you see very quickly when you look at the different genes, these are 4 different genes associated with inflammation, which is basically characteristic of the disease. What you see is that when the cells are put in when their cells are put in a standard Petri dish, there's very little expression of this. This is basically the Petri dishes, these gray bars. But when you put them in this biomechanically active system, then you have high expression of these different genes. Now, this shows that we actually have a system that models some aspects of the disease, and now we can actually start testing drugs on there. And here's a couple of drug candidates that we tested, and we started seeing some positive response about how it actually interacts with these cells. And we did this back in 2017, and the year after we published our paper, actually the results of a clinical trial that was happening in this field came out, which basically showed that there's a significant improvement when you add one of the drugs that we had tested. And what it showed is that in these 2 papers, one in PNAS and the other one in JAMA, that basically as you add these, as you have this drug, the mortality of the kids decreases tenfold. So normally they would die earlier, whereas in the treated case, they do not die as fast. And this is really exciting because it shows basically how these kinds of models that I mentioned can be used for actual real clinical examples, and that's very important. So now these kinds of systems are being more widely developed, and they're being used more and more in various kinds of medical applications, like being able to diagnose what kind of drugs are going to work for which people, and what kind of, for example, cancer therapeutics can work on which tumor types by being able to take biopsies from individuals and then tested their drugs on them. So this is all very exciting. The other thing that I would say is that this kind of area has a lot of I would say future. So I definitely think, and as you can see, it involves a lot of different disciplines. So being able to put good teams together, I think, is going to be very important. So having said a little bit about this area, I want to move to a second area, which is really about making the tools that enables these things. And this is about actually making the tissues themselves. So when you're making the tissues, particularly 3-dimensional tissues, it's very important to kind of recreate the right aspects of the microenvironment and the microarchitecture of the tissues. So we also worked quite a lot on developing technologies to be able to make them. And over the years, we worked on a number of different technologies to be able to do this. So, for example, there has been a lot of work on making 3-dimensional scaffolds. These would be degradable gels or plastic-like materials that can be used to seed cells on top, and they can basically grow and make tissues. There's a lot of adaptation of microelectronics technologies to this field, like being able to use micromolded structures, as well as lithographic approaches, and then apply it to these different cell and material systems to control their architecture. There's work on trying to use microfluidics, where you can basically flow cells and materials and cross-link the materials in particular shapes as they go past this microfluidic channel. There's also work on using fibers, self-assembly, and other techniques. For today, I'm going to tell you a little bit about our work on 3D bioprinting, which is basically applying 3D printers to the technologies like what we've shown here, which is applying cells and materials onto surfaces and being able to build 3-dimensional structures. So when you think about making tissues, there's a number of challenges that one has to face, particularly with this bioprinting aspects. And one of them is, how do we make the right kind of tissue complexity with the right three-dimensional architectures? How do we make the right kind of interactions between the different cell types? How do we make vascularized tissue? How do we prevent cells from dying? Particularly if you're going to put these tissues in a hypoxic environment like the body. And also, how do you make better printers themselves with better resolution and speed? So to do that, we worked on a number of these problems over the years. Early on, we've been developing tissues using different types of 3D printers. This is an example where we can deposit particular sacrificial materials, put the cells around it, and then be able to get rid of these sacrificial materials to create vessel-like structures that can be hollow, and you can flow liquids through them. And you can basically, what you can do is that you can see that they mimic the architecture of blood vessels. You can have cells that line them like endothelial cells. And you can have, when you do a cross section, you can basically have cells around these channels. And you can see that the cells closer to these channels, if this is the source of oxygen nutrients, remain more viable compared to cells that are further away from it. So this is how you can do this. But one of the things that's important to see is that there's limits to the size of these structures that we make. And the whole concept of bioprinting is basically to allow the cells to do a lot of the work in making the tissues. So the idea is that if we can put the cells in close enough proximity to each other, then they will, and basically add the right kind of molecules and compounds, then they will reorganize themselves over time and actually become more and more tissue-like. So this is after a couple of weeks, you see they look very different after a week, and initially they're basically very rounded. And these are some other images showing that, again, over time, we can get things that are better and better in terms of functionality. Now, in this field, there's a couple different areas that one needs to address. One is how do we actually make better materials in this field to use with the cells that we're printing? So this is what we call bioinks. These are materials that are used in the printers with the cells. And we want these materials to enhance cell function and cell survival. And also, to how do we make the printers better themselves? So make them faster with better resolution, and also be able to print many different types of materials. So we worked on both of these problems. I'm not really going to talk much about most of them, but I'm going to just mention that one of the things that we've done quite a bit is to make the right kind of inks or materials. And typically these are gel-like materials. And we've done a lot of work on making different types of gels that are now available commercially now. We've done a lot of work on making gels that are basically what we call universal inks that we can develop and print with many different types of molecules. We've made a lot of conductive inks and also oxygen generating inks. And all of these are important for different aspects of making tissues. Now, for today, with respect to improving the printers themselves, I'm just going to talk about one example, and that's specifically for making making printers that can print different types of materials. And why that's important is that tissues are highly complex, and they're made from many different types of cells and materials. So to be able to do that, to make them, you have to be able to print these different cells and tissues. Now, typically, 3D printing processes are limited to 1, maybe 2 to 6 materials if you want to get very fancy. And it's not really ideal for a lot of the complex printing systems that we want. So we wanted to focus on this problem. And a few years ago, we started working with our colleagues on trying to use nature as an inspiration to build these types of systems. So one inspiration is a spider. And what a spider does is that it has a number of different glands in its back. And it can actually combine the components of these different glands in different ways to be able to generate the silk of a particular property. and be able to then subsequently extrude this silk to out with a unique protein composition. Now, we can get this inspiration and build an engineering system that mimics it. So here's a system that basically has 6 different channels coming in. They're indicated by different colors, which also indicates that there's different materials potentially inside them. And there's white circles on top, and these are computer-controlled valves that can be opened and closed using the pneumatic approach that I mentioned earlier. And based on that, you can now start having these extruded materials come out in a very controllable way, so that you can actually see as you start extruding material, you can have control over what kind of things comes out. So this is an example of some of this. So imagine if you have 3 channels with red, green, and blue colors inside them. And you first open the first channel, then the second channel, then the third channel. Then the fiber that comes out will have the material composition of these channels coming out in a sequential way. If you open all the channels simultaneously, then all of these will be open in the same region of the channel. So by doing this, now you can actually have the complexity and coding controllability that's basically required in this process, and you can start having a lot of control over what comes out. Now, there's a lot of other things that one can do with this system. So for example, if you have the same red, blue, and green channels, and you basically open them in a way that as you open them, there's actually regions where multiple channels are open, then what you see is that you don't only change the chemical composition along the fiber, but you can also change the topography, because when you open multiple channels, there's more material and the diameter of the fibers increases. Also, you can play with other schemes and be able to control things like surface architecture of these fibers, or have multi-phase systems like air droplets, oil bubbles, or cells embedded in these types of systems. So this is all very important because it allows us to now put these upstream from nozzles in 3D printers to be able to create 3D printers that are basically have lots of control over what comes out. So this is a printer with 2 different inlets where we can control which one of these is expressed. And by doing that, you can actually start basically depositing these different materials. So you can deposit them in a layer by layer manner so that each layer has different types of material, or you can deposit them so that both of these materials are expressed in the same region of this printed structure. So by doing this, you can have lots of control over this, and we've advanced these printers to subsequent versions. So for example, here is a 3D printer with 7 different inlets, and you can actually start putting these in different combinations. So you can have basically combinations in which you have these individually, or binary combinations of these, or all kinds of other kinds of more and more complex combinations of them. So this really allows you to build hundreds or thousands of different combinations of materials, which is really exciting. And as we go forward, we think that these types of printers allows us to really build the complexity that's needed in a lot of the tissue engineering type processes. So down the road, we envision being able to really have controllable structures that are generated from non-invasive imaging of tissues, and then we can put different cells and different materials in different regions of these tissues, and then be able to have them function in desired ways. And obviously, for people who know about 3D printing, there's multiple different types of 3D printing. What we've shown in this example is a nozzle-based, so you actually print a nozzle to do that. But there's other types of 3D printers. For example, there are what we call stereolithographic printers. So this is an example of a stereolithographic printer, which is a digital micromirror device. And what happens here is that you can shine a laser onto a device that has a bunch of small mirrors inside it. And based on which of these mirrors are tilted the right way, you can basically get pixels that you can get projected onto a surface. And then this surface, because of if you have a material that's light sensitive, then the patterned laser light can actually induce cross-linking of this in a particular way. So normally this is done with just one type of material. By using the same kind of principles that I mentioned earlier, you can now have control over the different materials that gets used here. So here, for example, you can first cross-link one material, let's say this material in the shape of the star, and then bring in a second material as shown here, and then be able to change what the material that can get printed is, and then print a second material. And you can build these on layer by layer way. So imagine you have these systems here. You can see the cross section. You can basically build one layer, and then you can move the stage and then change the material and build a second layer and build systems like that. And really this allows us to now build a lot of different complexity in these systems and be able to control the architecture of these in a much better way. So maybe I'll stop here and leave some time for questions, but the general things that I want to get across are that basically when we think about personalized medicine, we think about areas like the omics world, genomics and bioinformatics and proteomics and things like that. But as we start kind of thinking about this, we have to kind of have a broader picture. So we think that there's a lot of engineering approaches like the ones that I mentioned today that can be used to supplement and enhance these types of systems. And they involve things like being able to make tissues and implants that are tailored to an individual, being able to recreate the individual's kind of biological behavior using these physiological models or organs on a chip models, making the right materials that can respond to an individual needs, and also devices that are smart and can have sensors embedded inside them. So with that, maybe actually I'll refer you all to our website. Anyone who's interested to learn more about the Institute or to apply to some of our positions, feel free to do that on the website. And also I'll end the talk with this particular picture, which is I like very much, which is a painting by Renee Mcgreet, which was a surrealist artist back around early 20th century. And what he shows here is that he's looking at the egg. It's a self-portrait of him. And basically be able to draw the bird that's emerging from it. So I think it's kind of really relevant for everyone's research, and I think that we should all, for everything that we do, we should look at the bird, basically see the bird in our early visualizations. So with that said, maybe I'll stop the talk and I'll be happy to answer anyone's questions.
Thank you very much, Professor Ali Qasim Singh, for a nice talk. It was really a interesting talk. And now the session is open for the questions. So if any panelists have any questions, please interact. And students, if you have any questions, you can post your questions in the chat box. To start with, there are some questions in the chat box, like for example, one student is asking like, what was the criteria for selecting the particular organ to start with, like, or the particular, for example, if you want to start with some organ, like what are the parameters we should know?
Sure. I think that's a really good question. So in most of the cases, I think the criteria is, one is what are the major organs that are important for your body and how it responds to different molecules. And really, liver is where a lot of the metabolism is done. So it's one of the most important organs. So that's one of the reasons we've been selecting liver as one of our models. And then the other tissue that is very important in our case has been the heart, because it's where we see a lot of the toxicity of different types of drug compounds. So those are a couple of ones that we've chosen, but really I think it's all based on the scientific question that one is asking, and being able to use the right model and the simplest model for the scientific question that you're trying to answer.
Yeah, thank you. So there's one more question, like, can we develop a personalized medicine for neurodegenerative diseases, like, what with approaches for the neuro diseases?
Yeah, so that's a really good question. Yeah, there's a lot of work in that area. You know, it's actually one of the ones that's very exciting. People are thinking about using things like brain organoids for making models of Parkinson's disease or Alzheimer's disease, or other types of diseases associated with the brain. And of course, you can use also peripheral nervous system cells to generate things like trauma or other types of things. So it's actually something that's been very actively being used, and I think that it's going to be one of the exciting things that may lead to new drugs or new therapies.
Okay. So one more question that is like, regarding the contrast agents in one of the slide you have shown. So what are some recent trends in technologies to have 3D scans of more complicated tissues or organ structure for 3D model development?
Oh, that's a
So
good
other than
question.
using
So
the contrast
um,
agents.
I yeah, so there's definitely yeah. So we've been using obviously a lot of standard CAT scans and things like that. We do put materials in the body, and we want to kind of be able to visualize them, and sometimes we do add contrast agents to the materials. I think when it comes to non-invasive analysis, there's a lot of new kinds of devices that are coming, of course, like the MRI and PET scan, and are kind of coming along. There's like techniques like second harmonic generation that you can kind of even get molecular signals out of it and be able to kind of look at things like collagen structures. But I think that it really depends on what is the kind of the resolution that's needed, what is the problem that one wants to address. In many cases, we've done just standard CT scans, and that has been fine for our models. But I think as based on different types of questions, different types of analysis may be required.
Right. And some more questions, like, for example, like, what kind of nanomaterials can be used, for example, like magnetic nanoparticles, and like how these kind of nanomaterials can be useful for these organ printing technologies or advancing this material
Yeah,
science?
so no, that's a good question. So, the way I think most of these can be incorporated into this field is really through designing the right kind of bioinks. So we've used different types of nanomaterials as both reservoirs for drugs, like nanoparticles that can encapsulate drugs and allow them to get released in a particular way. We've used nanoparticles as contrast agents in these types of inks. We've used nanoparticles to add conductive properties to these inks. We've used nanoparticles to modulate the rheological properties of these inks. So there's a lot of different ways in which they can be used. And I basically think that some of the parameters that you want to have with the nanomaterials that you put in, particularly for biological systems, is that you have to make sure that they're as robust as possible, and they're um, allowing the patient, or allowing the biological system that you're using to function undisturbed. So those are some of the things, but we do use a lot of nanoparticles in our systems.
And one more question, like what will be the long-term effects? So for example, if you use this kind of 3D printed tissue, like when you transplant into the human, so what kind of challenges? So will it perform really like a original organ, or so what are the challenges in this?
Yeah, well, obviously the goal is to make it like the original tissue. You know, I would say it when you're putting them in, if you're putting in the right cells and the right basically structure, then it could actually integrate fairly nicely with the host, with the patients, and this is really what the concept of transplantation is. If you don't, then there's other things that can happen. For example, you can have the immune system attacking some of these things, and that obviously sometimes is not good, but sometimes it actually having that inflammation induces some degree of remodeling and reorganization that's kind of good for healing as well. So there's different ways, but the ideal situation is that you want the tissue to be basically integrate into the person, and that's why you use cells that are, let's say, patient specific cells, so that the immune system sees the same cells that it sees everywhere else in the body.
Okay, and one more question in the same related to, like, what would be the shelf life of these organs like? So, is it really like
What is
a
the
original
what?
organ, or what kind of the shelf life of organ? This the 3D printed. What is the shelf life of organ? Like how much time it can be perform like organ,
Oh, shelf life.
or we
Okay,
shelf life?
gotcha.
Yeah.
Yeah, the shelf life. I think definitely if you're making things like big structures, you're going to have the same limits in the shelf life that you have with natural organs that you're transplanting. So, like, if you take an organ from someone and you want to transplant it to someone else, if you don't do a proper job, then you basically are going to be, the organ is going to die, and that's what's really important. So I would say the shelf life of larger tissues is, a day or two maximum. So you don't want to go beyond that because a lot of cells die. You typically put them in very low temperatures and all that stuff, or you try to keep them thing for cells like small tissues, then you can basically freeze it without problems and you can have longer shelf life. But for larger tissues, it's more difficult to freeze them.
And also any immune responses. Hopefully, it can be overcome by the, I think.
Yeah, I think the immune response aspect is really something that's dependent on if you're using the right cells or not. If you have the right cells, then you could potentially overcome the immune responses. Alternatively, you have to kind of have immune suppressants or other things if you're going to put it in people.
Okay, thank you. So maybe one or two more quick questions from a panelist. If anyone have any questions, please go ahead and you can unmute yourself.
Professor Ali, this is Vimal Srivastava. I hope you can hear me. Am I audible, Professor Gopi?
Yes, you're audible. Please go ahead.
Thank you for your nice talk. I have some inquisitive query, like what is the role of chemical engineers in devising such strategies for lab-on-chip approach? Certainly, we are doing something like rheology and microchannels, but those studies are generally either single phase studies, two phase studies. So how do you see these things growing? What is the role of chemical engineers out there?
Yeah, so sure. I think chemical engineering is a very important discipline that you things that are fundamental, right? So the fundamental things associated with transport phenomena, reaction kinetics, things that are important for many different types of problems, right? So I would say, first of all, a lot of people who made major progresses in this area, like researchers are chemical engineers, because they can not only understand the engineering, but also chemistry and biology associated with these things to be able to kind of link the different things. And I think there's the tool set that chemical engineers have is a very, I would say, complete tool set that to be able to apply to this field beyond just the technology, because you need to have insights about basically fluid transport and diffusion and shear stresses and also other things. So I think those are the things chemical engineering curriculum provides to this field.
Okay, thank you. Thank you, Professor Gopi.
Yeah. Yes. Yeah, there's one more question, Professor Pawan. You would like to?
Thank you. Thanks for the amazing talk, Professor Ali, and I really enjoyed it. I had one query regarding your crosstalk between organoids which you made in the microfluidic setup. And you were saying that it simulates more like the normal scenario in a body where organs will actually have a crosstalk. So I was actually interested in what were the results when you the crosstalk and when you allowed the crosstalk, and how much, because I think how much is good enough to simulate a real life scenario. I think this is doing some justice to the crosstalk, but till what extent should one go to simulate this crosstalk? And yeah, your comments on this.
Yeah. So just related to the second part, to what degree we should go, I think we should go to as simple a system as possible without sacrificing your results. So the outcomes. So being able to kind of understand what is the question and what are the things that are needed to be able to answer that question effectively. So what I typically think about is that we have to make sure that because you can get as complicated, you can try like every single tissue and attach it to each other and all that stuff, but that's not needed. If you have just a few different elements that are important, then that will get you quite a ways into having the right answer. So the other thing is, with respect to the first part of your question, I would say that we basically have been seeing crosstalk. So there is examples that we published in which you can have, let's say, if you have the liver and the heart interacting with each other, then you can actually, if you add a compound to the liver, if you add a compound that gets metabolized in the liver and generates like new compounds that are going to have secondary effects on the tissue, we can actually observe those things, things that you can't observe if you just have single tissue in the system. So I think that's another kind of exciting thing about this field, because you can actually start seeing basically multi-tissue interactions, which opens up a whole new area of research as well.
Okay. And do you allow pumping action between these things, or is it purely diffusion between? I mean, actually, because just
No,
to
actually,
understand,
it's kind of like circulation. Yeah, no, it's
okay.
like circulation. We actually have liquids pumping, like, between them. Yeah.
Okay, perfect. Yes, that's. I mean, that's a very nice example where the liver does something to it, and then there's a byproduct. So yeah, thank you very much. It was lovely interacting.
Okay. Thank you.
Thank you.
Any more questions from the panelist? There's one more question from the panelist, that is, if you're going to transplant a 3D printed organ in the children, so how will it be its growth with the body growth along with the? Oh, in the case of adult, it's fine, but in case of children, like.
That's an excellent question. No, that's an excellent question. In fact, that's one of the reasons why sometimes medical devices are not really good for children, because the children grow and the medical devices do not grow with the kids. So the concept of tissue engineering and having like fully biological is really designed in many cases best geared towards pediatric or children population because they grow, and some of these tissues can grow with the kids because once we put the tissue in the body, the synthetic components typically degrade, and you're left with fully biological components that can remodel and grow with the rest of the tissue. So that's one of the concepts why something like, let's say, heart valves that are made from tissue engineering are particularly attractive for pediatric population, because otherwise you have to change the heart valve every few years, and that's not ideal.
Okay. So in addition to the same question, like how we can avoid the material leaching, like is any material leaching, how we can avoid?
So I think it really depends on what the material is. Like if you're going based on materials degrading in the body, then definitely you're going to have some leaching. But if the leached product is benign and it's can be easily handled by the body, then it's fine. But if it's you leaches something that's toxic, then that's not good, right? So that's why the original material that you're putting in is very important, and you have to make sure that it's fairly compatible.
Okay, thank you very much. Like I think I taken a lot of time. And one final request, like you can share your advice to our research scholars. There are a lot of students listening to your talk and they're excited. And you can give some advice and suggestions, like
Sure, yeah, definitely.
how we can to add up, and or like
Yeah,
what kind of things you look
I
for.
definitely encourage everyone. Yeah, so just related to advice, I think that science things that you have to really be passionate about it, because it's not going to be as easy as some of other careers, and it can be an all-consuming thing. So it's a very open-ended profession, so you have to be very passionate about it. And I say to people that there's a combination of passion and perseverance that's very important. So one has to really understand how to persevere through difficult and be able to remain passionate about whatever it is you're doing and you want to be successful at. And then the other, there's a lot of little advice. You actually give a series of lectures or fireside chats that are, many of them are videotaped on our website at terasaki.org, that I encourage people to kind of also look into. We do have a fairly vibrant kind of lecture series that's about kind of advice about these types of things. So if people sign up, I'm sure there's going to be a lot of other sessions. It's a very big interest of mine to kind of give these types of advice.
Yeah, I have listened to several talks, like how to write a grant, and also like what are some of the things like. So it's really interesting.
That's right.
Yeah.
Right.
So
That's right.
Thank you very much. Thank you very much for accepting our request in a short notice. And also, I think we have taken your time in the morning. Like it's early morning there. It's almost like 6:30 or something. And thank you very much for the exciting talk, and also
it's
your
all
advice.
it's all good. I really thank you.
Hopefully,
really appreciate it, and
once the situation become normal,
I'm thankful.
Yeah, once the situation become normal, we'll invite you personally to visit our institute, and so that'll be a great opportunity for us also to collaborate. And on behalf of the IIT Roorkee and organizing committee, like I would extend my thanks, and see you soon at IIT campus whenever the situation become normal. Thank you very much, and thank you all the participants.
Thank you very much to everybody. Bye. Thank you. Bye-bye.